And I'm going to quit talking. All right. Karl, if you're ready to go, take it away.
Thank you. I appreciate it. Thank you, everybody, for your attendance to this webinar, and hope we get you sorted out with all the knowledge on potassium and water reports and the like, and we really appreciate it. I'm going to talk today about potassium nutrition, and I always feel that this is one of the most overlooked elements in a fertilizer plan, and I think you'll walk away with some expanded knowledge here at the end of 45 minutes or so to rewrite your agronomy plans from the ground up and kick 2022 off onto a good start. So just a little bit about me. That's my headshot. My official headshot. I used to use my wedding photo from 10 years ago, and corporate told me to stop doing that because I looked way too young. But I've been around in ag industry now for almost 8 years in Western ag, and I did my PhD in desert soil science. There's a few of us on this call with desert experience, and it's a good time. So I started out as an agronomist in the field, covering from Phoenix to San Diego, worked my way up into the Central Valley, and then a couple of years back, I joined as vice president of ag science here at Heliae, where I help work on biostimulant products and biostimulant technology. And I'm also, Jerome mentioned it earlier, I'm also the new chair for the Western Region CCA board. So if you haven't visited our website lately, I have the link there at the bottom. You can get caught up on all the latest news and membership and see who got the scholarships this year and all kinds of great resources, especially if you're going to study for the exams. We got a lot of resources there. So I'll kick it off with talking about what potassium is. And that's always a really nice place to get this thing going, because it's a really unique element in the fact that it's one of those elements that it's really hard to pinpoint where in the plant you're going to need that. And I'll contrast that. And the interesting thing about potassium is that the deficiency symptoms often look like other stuff, sodium burn and chloride burn, things like that. And so I think you'll see that and hopefully add some clarity to your crop scouting this summer. So potassium is one of the 3 major plant macronutrients, being phosphate and nitrogen being the other 2. It's very abundant in soil. And here I just pulled a fact from the Midwest here, about 12,000 pounds per acre at 6-inch depth in a sandy loam, and that's out of Nebraska. So there's a lot of potassium in the soil, but as you'll see here soon in this presentation, not a lot of it's available for plants. Most of it's tied up in mineral fractions. If you're a periodic table nerd, potassium was originally discovered and isolated in 1807 by Humphrey Davy, and he called it kalium. So that's why potassium has a K in front of it in terms of its nomenclature and on the periodic table, because its original name was kalium. And here's a fact for your next event you go to and you want to wow people with how smart you are. Pure potassium is extremely explosive if you just have it sitting out on a table. That's why we don't find just K, we find K+. The K+ form is not reactive, and that's very fortunate for us that when you put your potassium fertilizer out, your plants don't explode. And so if you want to look at pure potassium, you have to actually have it under wax. And here's pure potassium. It has a metallic sheen to it that's stored under wax, so it doesn't react with oxygen and explode. So I don't know what you're going to do with that fact for later, but just keep in mind, that's why we always see K+. K+ is a non-reactive form. It's the stuff that won't make your trees explode. So let's talk about the function of potassium in crops. And this is unique because potassium, if you want to think of a jack of all trades nutrient, potassium is it. Because potassium does so much for a plant that there's this whole laundry list I'm going to reveal for you. And if you want a sort of bottom line for your notes here, potassium influences the quantity and the quality of your crops. So yield, and then the quality metric of that yield, whether it's protein in wheat or sizing in nuts, however that specific industry measures quality, potassium has a direct impact on them. So let me pull up the laundry list here. So potassium helps with some plant functions, root growth, drought resistance, enzyme systems, helps with water relations in the plant, helps with photosynthesis, and helps with preventing energy loss. Inside the plant, and this is related to yield and to quality, is that potassium is key for translocating sugars and starch. So if you make your starch and sugars in those leaves, you got to move it to the crop piece of the plant. Potassium is how you get there. And so you can also help move other materials. You can help move starches. I mentioned that earlier. Increase your protein. Helps with lodging. If you go out next spring and your wheat's falling over, check the potassium status. There could be some issues there. And then finally, a healthy plant can resist crop diseases. So potassium plays a role in the disease management. So here is this laundry list. Just about every plant function is impacted by potassium. That's why it's needed so much. So what's the challenge here? One of the challenges with potassium is that it's very difficult to say, that's where you need the potassium. We can do that with nitrogen and phosphorus fairly easily. Here's the basic structure of an amino acid. And what you see on amino acid, this is the building blocks of proteins. Here's why you need nitrogen. It makes the amine group for a protein. If you don't have the amine group, you can't make protein. So that starts to limit your crop performance. I can pinpoint that nitrogen right to that functional unit there. Phosphate is also, you can pinpoint exactly where you need the phosphate. Let's just take DNA, for example, your genes. If you take DNA, you can see the nucleotide, it's right in the middle of the screen here. That nucleotide has a phosphate group on it. There it is. If you don't have phosphate, you can't make DNA or RNA. You can't make genes. If you have to pay for biological work, we all use ATP to do that, adenine triphosphate. Here's all the phosphates. If you don't have phosphate, you can't pay for biological work. So a really direct connection for nitrogen and phosphate. Potassium doesn't have that luxury. We can't go into these molecules and be like, oh, there's the potassium, there's the potassium. So that's why it takes, I think, a more thorough understanding of why we need potassium in our cropping systems, because we can't, observe it and connect it directly to plant function quite as well. Hence that long laundry list I just presented. So crops need K. So what I'm going to show here are some crop removal rates per unit of yield. So we'll start with grapes. 1 ton of grapes per acre, if that's your yield, will remove about 13 tons of potassium here in the oxide form, which is commonly reported for fertilizer units. If you look at alfalfa, you can see where these numbers jump even higher. 1 ton of alfalfa will remove about 50 tons of potassium off the field. Most of it will go to almonds as well. A half ton of almonds, 1,000 pounds of almonds will remove 85 pounds. So tremendous need. If you just compare the potassium need of almonds to the nitrogen need, you can see in a lot of these cases, the potassium need is right on par or greater than the nitrogen need. We pay so much attention to nitrogen fertility. We don't pay as much of attention to potassium despite this need. So here's grapes. I mentioned that 13 pounds an acre needed per ton. So you can take your per tonnage that you're pulling off your fields and multiply that by 13. And that's about how many units that crop needs that's going to take off the field that you need to replace at some point. So what happens if you don't meet those needs? Here's an example from the WRCCA app, and I'll mention this app again. It's just a good tool to have on your phone because you can look up nutrient deficiencies. This is what potassium nutrient deficiency looks like on grapes. Have these leathery leaves that have this yellow color interspersed through some green. And so next time you're out in the field and you see this, that's a good indication of low potassium. When we look at when those grapes need potassium, here's the crop stages. They're all on the x-axis here. And here's how many kilograms per hectare we need of that nutrient. If you look around coloring and then to harvest, look at this big potassium demand. It just skyrockets. And that's what we see a lot with these crops that produce such a strong sink, like a grape or an almond or other fruiting crops, is that once that fruit gets going, that's when the potassium demand really starts, and it often supersedes the nitrogen demand. So lots to pay attention to here on potassium. If we look at almonds, I mentioned that 1,000 pounds an acre needs about 85 pounds of potassium to meet that need. When we don't have enough potassium, the deficiency symptoms are often thought of, more of a drought system. You can get these curled leaves. It looks like the plant needs water, but really it needs some potassium. So that's where some of these tissue tests could come into play. If you let it get worse, sorry, I guess serendipitously, my bad circle that I just drew is also circling the new photo. Really bad potassium deficiency can also look like tip burn that's associated with salinity. So don't, a lot of folks start treating for chloride and sodium tip burn, and they're missing that potassium piece. So very, very different symptoms by crops, and often have symptoms that are confused with other problems in the field. One interesting thing about almonds is that their crop doesn't have this big skyrocket of a potassium demand like grapes, and I'll show you an example of cotton here in a second. Potassium demand in almonds is relatively linear, but what you have happen is that by middle June, you have about 70% of your potassium demand met that you need to meet for those trees. So you have a very small window to feed a lot of potassium to, starting here in about this March, April timeframe. You've got this very truncated window to get that potassium out, because you're kind of racing that clock here. So if you wait too long into the summer to put your potassium out, you're already missing about 70% of the race. So lots of focus has started to shift to early season potassium management because of the publication of these curves by the UC system. One of my favorite crops, cotton. 1 bale per acre of cotton will remove about 20 pounds of potassium an acre. So you can make plans for that. This is an example of, here on the top is early season potassium, and then what it progresses to later in the season. So oftentimes potassium deficiency is seen as maybe a magnesium or a zinc deficiency. And if it gets bad enough, I mean, a lot of folks would call this, some sort of sodium or chloride issue. So it's misdiagnosed often, and that's where these tools like WRCCA can really come into play, give you that photo library so you can match up to your symptoms to the nutrient deficiency and back that up with a tissue test. Here's cotton. Again, you can really see the skyscraper that starts around mid square and it ends just after peak bloom. But that rocket ship of potassium demand just goes up, and it's a very short window, but you've got to meet that demand. And if you actually track where the potassium's at in the plant parts, has a higher pounds per acre demand than nitrogen on cotton does. So lots of room for improvement in potassium management for cotton. If we look at silage corn, here's a typical yield, 30 tons an acre. Look at this whopping rate that the plants are pulling out, 219 pounds an acre in that silage corn. So a lot of material being exported that needs to be replaced at some point if you want to maintain the soil fertility. So just something to keep in mind. If you look at the demand rate, huge demand rate for potassium during that middle growth from around V6 into tassel. But if you look at the symptoms of potassium deficiency, they often look like salt burn, sodium and chloride burn. So it's often misdiagnosed. But this will impact your yield, silage corn, and even grain corn. And don't forget sweet corn. This potassium demand needs to be met. So we'll go into alfalfa. A big crop for us here out west. 1 ton of alfalfa will remove about 50 pounds of potassium an acre. If you don't meet those needs, here you start to get the early symptoms of potassium deficiency. So I won't judge you if you stick your head close to the computer screen to see this. This is a photo I took on a field that I had confirmed potassium deficiency. You see this little speckling of the leaves. That crop needs some potassium. If you let it go long enough, you really start to get some necrosis, some whitening of the tips of those leaves, and that tip eventually dying. So a good way to, all of alfalfa is based on how much biomass you can grow. And this is a sure way to lose yield and also lose quality of your hay. So I hope I've convinced you that crops need potassium, and every crop has a different sort of flavor profile for the potassium it needs, and what it needs to get by, and where that potassium is going to go, and timings in the season. So that's something that's worth having in your pocket as a certified crop advisor. But let's talk about potassium behavior in soils first, and then we'll talk about how the plant actually takes it up, and some issues that we can have from this translocation. So here's our system. I know this is a corn plant on the screen, but just pretend it's an almond or a grape or an alfalfa, whatever you want. Just use your head to put the right plant image there, because it's really all the same. We're going to start here with the soil, and then we're going to end on the plant uptake. And we're going to talk about the challenges that are all inherent to these systems with potassium management. So let's first talk about plant mobility and soil mobility. Here's potassium. It's mobile in the plant. And on this chart, they're saying it's not mobile in soil. However, potassium is highly leachable in sandy soil. So if you're on soils, they have a CEC of 3 or 4 or 5, you can actually leach out potassium and lose potassium fertility over time through your irrigation program. And I've seen this on very sandy soils. That very tip of the soil texture triangle, they can be irrigated into potassium deficiency. So this table says no, it's not mobile in soil, but just to write this down, depending on your source, highly leachable in sandy soils. So something to keep in mind. When we think about potassium pools in the soil, remember I mentioned back at the start of the webinar, about 12,000 pounds an acre of potassium in a 6-inch slice of Nebraska soil. Here's the bad news. Most of it is not really usable. 90 to 98% of that potassium is tied up in non-exchangeable mineral fractions. So it's the stuff that makes your feldspars, your micas, your geology. Any of you geology aficionados on the call will recognize all these names. If you're on the farming side, that stuff plants can't use that season. It's too locked up. It's too hard to use. We've got some potassium that's locked up into 2-to-1 crystal layers and clays. I'll talk about this fixed potassium here for at great detail because it's a challenge in California soils. And then we have what the plant can actually use. This tiny fraction, just look at the fraction of it, 1 to 2% exchangeable K on these clay and humus colloids, and then a tenth to a 20th of 1% here in soil solution, the stuff that's in the water. So not a lot to work with here. And this is what we're relying on for your plants to pull in. That's the small fraction you have to work with. Huge demand by the plant for potassium, we covered that just now, and a very small pool to pull from. That's why potassium management is so important. So when we go to do our soil testing and you get a ppm value on your potassium, you are looking at this tiny fraction and making decisions on the management. So again, there's a lot of potassium. If you did total potassium, you'd sort of blow your socks off. But when you take an actual soil test, we have a directed measurement to this small field, the small pools here. So let me show you an example just to revisualize of all the potassium in your field. That's this whole pie chart here. We're working with this small of pizza wedges here. That's what we're expecting to drive yields off of for our crops. And so very small soil solution and the stuff on clays and humic colloids. So that's why potassium management is so important. Huge demand, small supply. So that's the perfect reason to have a certified crop advisor come in and look at potassium management because we understand these relationships. One piece that's important is texture. And texture is important because what your soil value is telling you on your soil test needs to be correlated back to the texture of that field because the texture determines the release rate. Sandy soils can have a lower value on the soil test because they release pretty quickly. You can get by with a lower supply. A very heavy soil, this brown bar here, needs a much higher value to show up on the soil test to ensure an adequate potassium release because they like to hold on to potassium. They like to stick it to the clays, all that CEC, that cation exchange capacity, grab those ions and hold on to them, and the plant can't use them. So you have to have a higher value in order to make sure there's enough supply for the plants. So texture is a great way to think about potassium. Sandy soils can get by with a little bit lower of a test rating. Clay soils need a little bit higher just to manage around this CEC piece. One thing that comes up about Western agronomy is pH. And I know everyone's seen this graphic before. We're all familiar with, phosphate and being super tied up in alkaline pHs, and we've all heard that story. Potassium doesn't have that story. Potassium availability is not really tied up by pH in Western soils. It's more of a tropical issue here on the far left of the screen. But for most folks out west in the Western region CCA states that we cover, not a big problem. So it's not a pH issue. Really, it's much more complex. Potassium availability is related to soil water dynamics, local geology, soil texture, I just mentioned that, and how fast your soil is weathering or releasing those nutrients. So you went from having a fairly simple story with pH and phosphate to now having a much more complex story. So let's talk about how the plant manages through that complexity. So we have these couple of mechanisms for nutrient uptake, and we have mass flow, which is the movement of dissolved nutrients into a plant as that plant absorbs water. So think of it like a flowing river of water, and the water going up into the plant is bringing the nutrients with it. We have diffusion, which is nutrients moving from a high concentration gradient in the bulk of the soil into that root zone, and so the plant can pull it up. And then you have root interception, which is just like it sounds. The root actually grows into the nutrient, pulls it in. So potassium here, I've kind of compared and contrasted on this table. Most potassium uptake occurs through this diffusion mechanism. So you've got to have a good supply in order to drive that diffusion, because remember, diffusion is a passive process. You've got to have a gradient, low gradient at the root tip, higher gradient in the bulk soil. The bigger that gradient is, the more diffusion that can happen. So you've got to maintain healthy ppms and healthy values on your soil test to make sure diffusion is happening below ground. And then potassium is also pulled in through the water stream on mass flow, and that could be important as well. So if your soils are dry, you won't be able to move any potassium up into the plant. So challenges here, potassium, you need a lot of it. There's not a lot to go around. And here you can be impacted by multiple uptake pathways. So if you think about mass flow, think about water supply, how good is the irrigation management? Or if you're from dryland country, how much rainfall have you had lately? If you think about diffusion, think about a supply management. How healthy is my soil? How well is my fertilizer plan doing to keep that diffusion gradient healthy? And then root interception, just like it sounds, plant root grows right into the nutrient and pulls it in. So let's talk about the challenges. We mentioned how that plants need potassium. Here's how they get it, but what gets in the way? And that's this just challenge of potassium antagonisms and fixation. So I'll go through that here over the next couple of slides. One piece I'd like you to write down, because we are most likely headed into an extended drought, and sodium challenges are expected to rise because we have declines in water quality. We don't have as much water to leach. We don't have as much rainfall to leach. So sodium values are going to start creeping up on our soil records. I'd bet money on it. $3. I am an agronomist, not a lot, I don't have the big bucks. But here we have this issue with potassium and sodium, where sodium directly decreases that plant's ability to pull in the potassium. There's an antagonism. Sodium antagonizes potassium. And here's why, in case you need more facts for the weekend. Sodium causes poor uptake of potassium. And that's because you have this issue with membrane depolarization. It makes it hard for those plant roots to pull up the potassium with the inward rectifying channels, or KIRs, if you really want to have a cool acronym. So the plant can't pull it up. The plant has a hard time pulling up potassium in the presence of sodium because of this depolarization issue. And then on the other hand, you get increased potassium leakage out of those roots. So the plant can't pull it in, and then it starts leaking it. So it's bad news bears all around. The sodium increases K leakage because it's activating these outward rectifying channels, or KORs, And so you're getting kind of punched in the face twice. You can't take the potassium up and you start leaking it. You can have a sodium issue and go down the tubes in your potassium management very quickly. There's a great paper on this subject, Wakil 2013, that describes this in length. And good salinity management usually pays off. Here's a way to make your salinity management program help your potassium program overall. So I always say, I think I'm going to make a t-shirt that says this: deal with saline sodic soils early and often. Don't wait. As soon as you start getting those signals of sodium creep or chloride creep, make a plan to get rid of it. And then use your data streams to help you diagnose those challenges and try to forecast issues before you get into this uptake and leakage issue with your potassium program. So here's another way to think about antagonisms, and this is the Mulder's chart. We've all seen this before, where it's connecting all these different dots. And these green arrows here are showing that in the presence of excess nutrient, you have a decrease in the uptake of another nutrient. So we'll just start at the top here. If you have a whole lot of calcium, that can cause an antagonism to potassium uptake. So when you look at those soil reports, you can reveal some of these things, like if you have values that are very out of whack. So I'll sum them up here. Calcium, phosphate, nitrogen, magnesium, and sodium. I know I'm kicking the dead horse here with sodium, but it's super important, can decrease K uptake. So if you have a soil that's a little bit out of whack, that can feed into your potassium management. If you've got a whole lot of potassium, you can decrease your boron uptake. So it works both ways. Antagonisms can happen on either side. Manganese and potassium are synergistic. And then you have uptake in iron when you have good potassium values. So lots of information on this chart to take a look at. Here's the summary, but just a lot of things to think about when you're managing your soils, when you're managing water, you're managing your plant tissue reports. There's lots going on, and I think that's what makes certified crop advisors the best in the business, because we can consider all these different nuanced interactions. So let's get into K fixation. I ran into this issue early on in my career in agronomy down in Riverside, California, and I couldn't figure out what was going on. I had a potassium program, and I was aggressive with my potassium program. We were making investments into the citrus program, but I was still coming up short on these potassium values, and I couldn't figure it out. So I went down this rabbit hole of research, and that's where I dawned on me this K fixation piece, where we have this issue with the soil clays themselves can actually store your fertilizer program. Here, I'll just point an arrow to it. Your potassium that's coming from your fertilizer, they can compete for it and store it away in these little Oreo cookies here, where the potassium is now not available for plants. So they can fix the potassium physically within their structure, and the potassium is not available for plants, despite your aggressive program that you put out. So we have these vermiculite clays. They're pretty bad at it. These kaolinites, if that's what your soil is made out of, not a challenge, because they're not putting it in a sandwich. This can be released pretty quickly. Your smectites, clays, they have a big space here where they can store lots of potassium and fix it and keep it away. So you've really got to pay attention to the geology of your soil. And I'll show you how nuanced this is, because I know there's someone on this call who's been aggressive with their fall potassium dry program. They have an aggressive liquid program in season, maybe a foliar, and they're still coming up short on potassium. K fixation could be the way, the reason why this is happening. So let's talk about K fixation. Here's the geological map of California. So I was in Riverside here with my issues, and that's where I discovered that these soils I was working on are all derived from this igneous intrusive, this periwinkle color here, all igneous intrusive rock. So where we have K fixation is where I just circled around this blob down by Riverside, and then you have challenges here in the Central Valley of K fixation. It all has to do with where the soil came from. If it came from the alluvial material, leaving the Sierra Nevadas made out of this igneous intrusive material, that sets it up to be prone to K fixation. And there's a great body of literature on this out in the this IP and I Better Crops magazine from a few years back. So highest K fixation capacity forms on this igneous intrusive bedrock and alluvium. So just track your field's proximity to some of this this periwinkle colored map, and you'll probably have some K fixations in the area. And then there's also a texture piece. Young, coarser ground that contain vermiculite. So there's three categories here. These fractions are prone to K fixation, and the soil's got to be a little bit younger in age and have that vermiculite. Let me go back one slide. And this piece here, this vermiculite, the super smushed Oreo cookie, that can really cause problems with K fixation. So lots of challenges here, lots to keep track of, but this explained a lot to me. And once I figured this piece out, then I could really start managing my potassium, and I eventually CCA'd my way around the problem. So let's talk about K fixation spatially. Here's a map of the Lodi area, and they've mapped out these different flavors of soil here, region 1, 2, 3, 4, and 5. And just to show how subtle this changes between K fixation, here we have this pink, it has high K fixation capacity. So it's sucking up all your potassium and not letting it go for the plant. But you go just a few miles down the road and you have a soil that doesn't have that capacity because it's slightly different in its geological setting that it formed in. And then just down the road, you have this moderate change. So very small changes in space here in the Lodi region give you very different changes in the K fixation capacity of the soil. So what do these soils have in common? I mentioned this this pink soil here, the region 3. There's region 3, high K fixation capacity. I mean, here's the age. It's young. It's full of this these minerals that can pull in the K and not let it go. And here it's, so you can see its relative K fixation potential is high. So not a lot of change in that area, just miles wise, but huge changes in that soil's ability to fix K. So super challenging. And the UC system's done some work where they've had to fertilize their way out of these high K fixation soils. And it takes like 1,500 pounds an acre of sulfate of potash banded in order to saturate the inside of that Oreo cookie and make it so that those clays can't tie up any more potassium. This is an example on cotton, but it just goes to show how troublesome that the K fixation can be. And then we also have some research that shows that K fixation can increase with depth. So these bars that are this pink, this pastel color on top, that just shows how that K fixation capacity increases as you go down in the soil. So you've got this spatial extent across the miles and counties, and then you've got some K fixation changes over depth. So lots of challenges here for K fixation. So how do we understand K fixation in California? If what I've said is ringing a bell, like my bell got rung a while back, let's talk about what we can look at. We can look at soil test results, and one misleading thing on K fixation is that your potassium values on your soil test will still show up high because you're pulling that potassium out of the Oreo cookies. Even though your plant can't use it, the test is pulling it out. And so you get kind of a false positive here. So I'm showing you a soil that had a optimum value of potassium. So you might not want to invest in K fertilizer, but when you have the fixation issue, it really reveals itself in the tissue samples. So that's where combining your soil and tissue test results can really point you in the right direction. Do I have an uptake issue with my potassium? And then the soil series information, if you go into your soil series, you can actually read what it's derived from. If it says igneous alluvial material, you might have a K fixation issue. Here's an example of that. Here's that same soil in Riverside that threw me off so bad. A Hanford coarse sandy loam, beautiful soil if you're a soil nerd like me. But look at the parent material, alluvium derived from granite. That's that igneous material I mentioned earlier. And that's what made, matching up all my reading and research, that's what tipped me off. Like, oh, I got a K fixation issue, because not many labs measure that. It's a difficult test to measure K fixation capacity. So you're gonna have to piece your detective work together from other sources. And if you really want to nerd out, you can pull the geological quadrant for your field and read exactly what your geology is, exactly what your soil is derived from, what age it is, what the mineral type is. Usually you can stop at understanding your soil series information, but if you really want to go into the quadrants, there you can do that as well. I suggest, if you think you have K fixation, to look up this article. It came out 10 years ago now, believe it or not. Potassium fixation and its significance for California crop production. All the little anecdotes about how challenging this is, and the fertilizer pounds needed to saturate the clay layers are all coming from this article. And it just goes to show how problematic this is, and how some areas escaped K fixation just from their geology and geological history, and some of them deal with it now every day, and that could really help guide you to some management decisions and a better way to understand it. So I'll go through some K dynamics now that we talked about K fixation, but we're going to talk mostly about the plants. I showed you that plants need potassium and it changes over time. One piece to think about with plant transport is that it's carried through the xylem and the phloem, flows up into the plant. And the plants that transpire less accumulate less K. So you need to have a good K supply and get it into those fruiting bodies or nuts or the reproductive parts. Luckily, potassium is mobile in the plant. So if you are a little short in your kernel, it can move it from the other tissues. It's not like calcium where you're fixed. Plant K is relatively mobile. Dry soils mess up your potassium nutrition because the K solubility is reduced, and water and salt stress can induce a potassium deficiency. If you're having a hard time maintaining that wetting front across your soil depth where your roots are, the plant won't be able to pull in the potassium because there's not enough mass flow, remember, going through the water stream, and you could have the presence of excess sodium, and that I mentioned earlier can make for increased K leakage and give that plant trouble for pulling the potassium in. And then finally, drip systems. If you have a lot of roots in one spot under the drip system, they can locally deplete their potassium. So if you do some of those tests where you're right at the emitter, you could have shockingly low values of potassium because your plants have taken it all out. And that's where the water is, that's where the root's going. So you've got to maintain that wetting front and make sure you have the high potassium. You can take a soil sample, just a foot over from the emitter in some of these soils and find all the potassium you want, but you won't find any roots. So that can be an issue, especially under our permanent cropping systems during the middle of the summer, where those plants are growing right where the water is. So some key points: soil and plant tissue results should agree with each other. Good supply in the soil should give you a good supply in the plant when you go to take your tissue report. So optimum values, that's what we're trying to manage for. The mismatch, if they're not matched up, either poor supply on the tissue, poor supply on the soil. I mentioned earlier that K fixation, where you can have a whole huge number on your soil report, but hardly anything in the plant. There's something going on. It's fixation. There's a supply challenge with the fertilizer program, or maybe there's an uptake problem. Like I mentioned, that concentration of plant roots that's locally depleting your potassium and giving you that signal. So the results help you investigate, how do I solve this potassium deficiency issue? And then just remember that critical values are lab crop state specific. So it's all kind of customized to the diversity of crops that we look at out west. So here's an example. I mentioned this earlier of how I ran into this K fixation problem, where I had a high value on my soil test, 253 parts per million, almost in the very high range. I'll call it optimum, just to put a positive spin here. But my tissue report was coming back super low, like almost deficient. And that's where I had to do this line of investigation and really kind of put my CCA training and knowledge to the test. That's where I discovered I had a potassium fixation issue. So what we did is we started banding higher rates of SOP right along the irrigation lines in the fall, so we could saturate the clays and free up more potassium in the long term for the plant. So we turned this franchise around. That K value in the tissue jumped back up, and we were able to work our way around it. So I'll sum up here. Just how we can relate to potassium. Large soil test deficiencies and K fixation can be solved at pre-plant or with that fall dry fertilizer shot we do. That's one of the best ways to do it. Get the most bang for your buck. In-season demands, of course, can be met with fertigation and foliar applications. Tissues and soil sampling help you forecast nutrient demands and help you solve problems. And really the timing, which is super cool. If you think about variable rate, we have variable rates in the timing of what crops are need. You can dial in some really nice programs in season if you pull those K demand curves. Like I showed the one for grapes, I showed the ones for cotton, and really make a difference to your grower. And of course, if you're running into that soil type and K fixation sort of conundrum, that's where diving into some of the soil series descriptions and geological quadrants can really help you out. So I have some apps for you just to write down. The IP&I has a nice calculator that you can go to this link, and it'll help you forecast. Here, alfalfa 6 tons an acre a year. This is how much I'm going to remove. So you can plan for your nutrient demands from crop to crop. And so they've got it just about every crop, and it's definitely worth a look. The Soil Web app, you can put it on your phone, and you can look at when you're in the field, what is this stuff made out of granite or what? And you can actually make that determination right in front of your grower. And then the WRCCA app, if you Google WRCCA into the Apple or Android store, you'll be literally walking around with a library of photos of potassium deficiency, all kinds of different crops. Very useful tool. Thank you to Yara folks for putting that together. I highly suggest you put that in. And these are all free, so even better. So with that, Jerome, I'll open it up for any Q&A and we'll go from there.
Thanks, Karl. I don't really see anything in the questions and answer session at this point. We did get a correction from somebody in the audience, just a little bit of a minor issue, but nothing to worry about. We do have a question about, have you seen ever a situation where you can get a potassium toxicity, like excess potassium, like extreme levels of potassium? Is that something that you've ever run into?
That's a great question. So I came up as an agronomist in the desert, and we have some soils down here that have 900 parts per million potassium on the soil test, and we don't see toxicities from the potassium, all that potassium. We don't see any issues. I think it'd be very difficult to drive a potassium deficiency. I think what you might see is maybe a potassium antagonism. Right. Where you have that much potassium, maybe it's interfering with your nitrogen program at that point. But
or also we've heard about magnesium antagonism, getting magnesium and potassium out of balance.
Yep. All right.
Matt. Just a quick question for those people I forgot to mention at the very beginning of our talk. CCA hours will be posted at the very end. You'll be able to get your credits. This is to make sure that you don't, you stay through to the very end of the thing. We will pop up the QR code. So make sure you have your CCA app on your phone ready to go. And you basically take a picture of the QR code and you'll get instant credit for our presentation today. So you'll get your 2 hours that way. And so that's pretty cool. Let's see, what else do we have here? So we have another question here. When you're banning fertilizers in a vineyard, are you putting the full amount or just a percentage on basically the question of banding? So if you need 200 pounds, would you put all the 200 pounds in a band? Or, do you use a portion of that and try to say, if I need 200 pounds total, am I only putting, say, like 67 or, maybe 80 or something like that in that band?
Yeah, that's a great question. So if I were splitting my allocation up, I need this many pounds an acre of potassium to make my crop. then I suggest if you are going to ban it, you don't ban the full 100% because that potassium, you might run into fixation issues. If you're on very sandy soils, you might leach it out of the rooting zone, which can be problematic. So if you do need to go up front a little heavier with your dry application, and it's going to sit there for a while before the plants can pull it in, you might use another tool like a humic acid to hold onto that potassium in the soil, keep it in the root zone, and kind of have a little bit of peace of mind that the potassium is going to be there when you need it. And then you can come back in, with the other 30% in season with your liquid program. So, and foliar program. I think that's great nutrient management.
Sounds good. I have another good question. Are there any remote sensing tools that you're aware of that can give you an idea where potassium deficiencies are occurring in your field, say from a drone or a fixed wing aircraft, something like that?
There's plenty of drone and satellite data and other imaging products that are out there that can show you where plants are less vigorous or more stressed relative to other areas of the field. But I don't think they're sensitive enough to say it's because of potassium. I mean, it really kind of just gives you the map to the treasure chest, like, hey, go check out this area, figure out why it's showing so much more stress relative to this other healthy spot on the field. And it might be potassium, but it could also be, clogged drip emitters, and the gophers chewed up the sprinkler system and all these other things, so that really, I don't think we're quite there yet to pull specific signals for potassium. I think you still got to be out in the field scouting and doing your due diligence on why am I seeing this pattern of stress.
I would agree with that. Absolutely. Another interesting, are you familiar with the quick release test for potassium that has been done? I think it's the old Unical test.
No, Drum, I'm not. Do you want to elaborate on that?
Yeah, basically, I think Unical came up with a sort of a liquid incubation test. So they take your soil and wet it, and then they let it incubate. And it's like a K release test, I think is what it is. And it gives you just sort of a relative index of, how much potassium under nearly saturated solution conditions that would end up in the soil solution, and it kind of shows you how many soils are thrifty with potassium, won't let it go, or how many can actually release a significant portion of potassium into the soil solution and meet plant needs. So that's sort of an incubation study that can be done. It takes time, and it is a little bit expensive because of all the handling that's required. It doesn't get a lot of use, but it is available in certain situations. But I know that Stu Pettygrove did some work on the release test for basically K fixation. And I think digging into Stu Pettygrove's paper would probably give you more information on how they figured out what those fixation potentials are. But certainly the younger soil types will fix more than others.
That's a good point about the soluble calcium versus the soluble potassium versus the exchangeable potassium, that having that extra little bit of information. So the quick release test, you can also do a saturated paste test, something that'll tell you how much actually soluble potassium in the soil water there is versus what's in the clays. Those are two really good bits of information to have for managing your potassium, because you can have high, ppm of exchangeable potassium, but if it's not getting into the soil solution, then the plant can't pull it up, and you can have a deficiency despite a high test. That's a good point.
The threshold I've heard for if in the saturated paste extract is about 0.3 milliequivalents per liter, I think is you want to have above that to supply decent amounts of potassium to the plant. So usually that's the bottom level, but frequently we see soils on the east side, San Joaquin Valley, derived from granitic parent material that can be 0.1, or less. So yeah, definitely, I think that 0.3 is really a good one. We've got another time for a couple more questions. How about the different sources of potassium fertilizers? In terms of availability, does the associated anion with the potassium have any impact on availability?
Jerome, would you mind repeating that question?
Yeah, okay, so we've got different sources. There's potassium sulfate, potassium chloride, potassium thiosulfate, potassium nitrate. Do you feel like the availability of potassium from those different sources matters?
Yeah, that's a great question. So all those different flavors of potassium carry differing potassium loads with it. So sulfate of potash is 0,050, muriate of potash is 0,060. So you get different percentages, and that'll affect your fertilizer math. Really what you got to worry about is not so much what the potassium is doing, but its partner, the anion, the negatively charged. And I'll use those two examples. Muriate of potash is potassium chloride. So if you are in an area that's already prone to poor water quality and salt accumulation of soils, you might want to avoid that source because the chloride concerns that come with it. Sulfate of potash, it's potassium sulfate. So that sulfate could be a better solution for you because you don't have to worry about any chloride burn using a sulfate-based product. Yeah, this is a really good discussion. Usually the potassium quality doesn't change a whole lot. It's the same K plus, as long as it's not exploding, because it's pure potassium. Really, it's just the properties of that anion you need to worry about and make your decision from there.
Sure. Another question here looks pretty interesting. So if you have excess sodium in your soil, can you actually offset that with a potassium application? So like a fertilizer application of potassium through, say, a drip system or something like that, a fertigation of potassium, would that reduce sodium problems?
I don't think so. I think you'd be better off just optimizing your input dollars by dealing with the symptoms of the soil that usually lead to sodium accumulation. And that's often poor structure. When those soils fall apart because there's so much sodium in it, they stop draining and you start getting excess sodium and usually chloride. So I think you're instead of trying to combat that with potassium, you'd be better off on a sodium and salinity reclamation program, opening that soil up, leaching out the sodium, and solving it that way. I think it'd be a better use of those dollars if you have a salt problem.
Yeah, and I jump in here and just say that I think we would take advantage of what they call the lyotropic series, or the kind of which cations can exchange other cations and knock them off clays, and the big fat calcium, with two positive charges is pretty good at knocking off sodium, which is very loosely bound or held to the cation exchange, whereas the calcium is more strongly held. And then, and potassium being also single charge positive, if it's in excess, can actually start to behave a little bit like sodium too and cause problems, although you rarely see concentrations at a high enough level to cause that type of issue. But, certainly magnesium we've found, is much, much smaller than that calcium. And usually we consider it to be beneficial, but because of its smaller ionic radius, the magnesium can sometimes, if it's in excess and calcium is too low, can cause soil problems as well. But yeah, I think that you're right. I would go in with more of a gypsum or a calcium liquid application if I had a sodium problem. Excess potassium is going to be a pretty expensive way and ineffective, I would say, to correct that sodium problem for sure. Cool. Where are we at on time? Let's see. We are getting right up to our 2 o'clock hour, so I think we're just going to call it quits on that. And thank you, everyone, for a really good discussion in the Q&A. So we're going to move on to Aaron's presentation, and I'd like to thank you for offering to present today, and just sure glad to have you as a new board member. Pretty exciting.
Oh, thank you.
Just
I'm excited to be here.
Yeah, really neat that with one of the upsides of COVID, if you can think of anything that's good about it, which I can't, but the one thing I can think of is the fact that it's forced us all to start using these video type tools more. And it allows people who are far away from Sacramento, where a lot of our CCA activity takes place. It allows Karl, who lives in Phoenix, to be our chair, allows Aaron to get involved. And so we're able to pull from a lot wider range of people and expertise. And it's great to have you on the board, Aaron. So
thank you.
without further ado, we'll have Erin talk about how nutrition and crop disease and crop pest problems interact. And I've always felt that good nutrition can solve a lot of problems or prevent a lot of problems with those types of issues. And you don't necessarily have to go straight to a pesticide. So take it away, Erin.
Yeah, well, thank you for that introduction. And Karl, your presentation was great, and it really leads well into what I'm talking about as well. There's a lot of overlap there. And so, yeah, my name is Erin Wingate. I'm a certified crop advisor in Ventura County, and I work for Tritech Ag Products. And our company is a pesticide sales and application company that got into fertilizer about 10 years ago. And we decided to go that direction because our PCAs found that their program could be great, their timing with spray and spray execution could be really good. But if the grower's fertilizer or irrigation wasn't well done and planned out, and the timing of water and fertilizer were off, then the pest problems would be severe, and it'd be really difficult to fight that with pesticides alone. And so they wanted to start getting more involved with fertilizer and give recommendations so that the crops would be really strong before having to use the pesticides. So it's so much like human health. You know, the first thing that we need to do is get lots of sleep, take vitamins, eat well, exercise. And for the plants, it's very similar. They need the right ratios, right amounts, and right timing of all of the nutrients in order to build up their own chemical and physical defenses. They also need a healthy microbiome, just like we need a healthy gut microbiome. And they need the nutrients in the right ratios and the right amounts for different growth stages without putting down so much fertilizer early on that you might get a salt problem or make the salinity issue worse. So for example, if you were trying to treat your sodium problem by applying potassium, you would end up increasing the salinity of the soil solution without really taking care of the sodium problem, because the potassium isn't able to knock sodium off the exchange sites as effectively as calcium would. So making sure not to over fertilize is really important. So the kinds of defense mechanisms that we want the plants to be able to do for themselves is mainly building strong cell walls. So the structure of the cell wall needs to be intact, and it has to have an integrity so that there aren't gaps in it or issues with growth that made it deformed. The cell walls also need to be thick and flexible and strong. And then plants also make different kinds of chemical deterrents that signal to the pests or pathogens to go away. And then there's also lignin, which is really difficult for insects to chew through, or for pathogenic fungi to get through. And then also we want the plants to be regulating their growth rate. So we don't want too much vegetative growth because that could attract pests. But then we do want fast root growth at the beginning of the season so that the crop can access every nutrient that it needs, get all the water it needs, and also kind of outgrow pathogens. And we'll talk more about that. So I'm going to go through the macro and micronutrients in detail and show how each of those affects crop disease prevention. So here are some macronutrients, and this is just a few examples of how they're used in plants. So like Karl showed earlier, nitrogen is a component of amino acids. So you need lots of nitrogen in order to build proteins. And nitrogen is the most limiting nutrient to crop production usually. Phosphorus can be right up there as well in certain systems. And phosphorus is also quite limiting in natural ecosystems. Nitrogen is also used for the cell's energy currency, ATP. Phosphorus, same thing, required for ATP, and it's in DNA and RNA. And it's required for tissue growth. So there's lots of phosphoproteins and phospholipids. So you need it in order to build cellular structures. Potassium is important for cell wall thickness and stomatal regulation. And it's involved in metabolism in other ways. It's important for electron transport. Calcium, we're all pretty familiar with calcium for its importance to cell wall structure, cell division, and elongation. But it's also important for nitrate uptake. So having calcium available can increase nitrate uptake, and that can have effects further down the line on amino acid production. And then also calcium and potassium as well are involved in carbohydrate and nutrient translocation. So once we have photosynthesis going really well and we're producing carbohydrates and we've got these nutrient stores, then all of that needs to be transported to new growth or to developing fruit or seed. Then finally, magnesium. It's at the center of chlorophyll, and it's required for a lot of enzyme activation, especially things involving phosphate. So it's required for ATP to work properly. So then how do all of these things affect pests and pathogens? Because those were all of the ways that nutrients function in the plant. But then all of that is not only important for growth, it's also important for pest prevention. So nitrogen can affect pests and pathogens both in excess and in deficiency. If you have too much nitrogen, then you can build up amino acids inside of cells, and then it can leak onto the leaf surface. And this is a common theme in a lot of these nutrients, because there are multiple ways you could end up accumulating excess amino acids or carbohydrates. And when you have too much of that that hasn't been assimilated into structures, then it's going to be like oozing out of the leaves in very tiny amounts, but that's enough to attract and feed pathogenic organisms. And then what makes it worse is once you have that leakage, if you will have high nitrate or high nitrogen, you're also growing a higher proportion of tender young vegetative growth. So you can imagine you apply a whole lot of AN20 and then boom, you get a lot of new growth. And all of that is very soft and delicate, and the pests, just like the insects, love it. And so then you've got this plant that is very vulnerable. So you can end up with a pest issue that escalates quickly. And then at the same time, when the plant has a lot of nitrogen, it kind of feels like things are good. You know, we're all fat and happy. And so the plant lowers its defenses and it produces less phenolics and lignin. And those would be deterrents. So we want to strike this balance, right, where we're applying enough nitrogen so that we've got a vigorous enough growth, but not so much that we're lowering defenses and making the crop very attractive to pests. But then nitrogen deficiency, that can be a bad one too. And I think most people are kind of more familiar with that. If you don't apply enough nitrogen, you see the stunted yellow plants. They seem so weak that they can't fight off any disease. It also prolongs early development, so they're in that early vulnerable growth stage for longer. And then if the nitrogen deficiency occurs later in the season, then nitrogen is mobile in the plant. So it's going to be pulling nitrogen out of the older leaves and into the new growth. And so those bottom leaves will start to senesce, and that dead decaying tissue will attract facultative parasites like Fusarium, Alternaria, and other things. So both ways are not so good. So test the soil, test the leaf tissue, and walk the field, and you'll be okay. Okay, so phosphorus is another important one here. And it can be tricky because in our Western soils, availability can really be limited. The main way that phosphorus affects disease management is through its effects on building a healthy root structure. If you can build up healthy roots really, really quickly, then it'll help the plant sort of grow out of or escape a soil-borne disease. And, take this with a grain of salt, because we all know that soil-borne diseases can be like very virulent, and simply applying phosphorus is not always going to solve that problem. But we at least want to give the crops a fighting chance, and making sure we have an adequate supply is really helpful to that, especially if disease pressure is low to moderate. Also test the soil for disease presence so that you know what you're dealing with. So having enough available phosphorus will help those roots grow a big network, and then that causes a cascade of other good effects. So the healthy root system helps the crop access more nutrients and water so it can mine a bigger portion of the soil and have access to the other micronutrients. Applying too much phosphorus at once can limit zinc availability and calcium to a certain extent, because phosphorus precipitates out with other cations. So in this picture, this is just showing that we have salt affected soils here. We can get really cloddy, high clay, things like poor structure, compacted soil, heavy clay can all make it difficult to grow a healthy root system. So I got this information from California Fertilizer Guidelines, and this was put together by Daniel Geisler. And he has different crop responses to Olsen test phosphorus. So you can see that they differ somewhat, but for the most part, you want to see your Olsen P above 40 ppm. And phosphorus availability depends on pH. The best is 6 to 7 or 7.5. And it's also very affected by soil moisture, temperature, and like I said, soil texture and structure. Mycorrhizal fungi can also help the crop mine phosphorus from the soil. The mycorrhizae extend the crop's root network, and they can go out and mine phosphorus, bring it back to the plant in exchange for photosynthate. So maintaining good soil microbiology by providing a carbon source or doing anything you can to add organic matter or increase organic matter will help build up a healthy microbiome. So next we're going to start going into the cations, and potassium we already heard a whole lot about. So just like Karl said, it's important for all kinds of metabolic functions, cell wall thickness and strength, and it's involved in enzyme functions. It's necessary to produce proteins, starch, and cellulose. And if you don't have enough potassium, then the plant isn't able to assimilate amino acids and sugars into those larger protein and starch molecules and cellulose. And so you end up with a buildup of the amino acids, and that can accumulate and leak, just like nitrogen deficiency can cause leakage, so can potassium. And so potassium fertilization has been shown to decrease the incidence of foliar diseases like leaf blight, leaf spot, stem rot, things like that. And so you can apply it foliarly as well as to soil, and both have their advantages in different situations. Calcium, it works alongside potassium in building up the cell wall and membranes, and deficiency can cause, like you see in the picture, that's celery black heart. And although it looks like it could be some vascular fungal infection, it's really initiated by malformation. The plant wasn't able to build its structures properly. And so you get some senescence. And then once you have open wounds, that leaves the door open for a secondary disease to come in. So what starts out as a structural problem becomes a pathogen issue. And so you can have similar things happen in apples, tomatoes, bell peppers. And you can also see signs of calcium deficiencies in leaves. In our Western soils, we have lots of calcium in the water and in the soil. Some of that can get tied up because the pH is quite high. But also we can get calcium deficiency just due to weather. And that can be an issue when we have a lot of overcast days, and when evapotranspiration is low. That's when you want to watch out for this kind of thing. So making sure that you fertilize with calcium ahead of time, and try different foliar sprays and see if any of those are effective to your crop. Making sure to spray them on to that younger tissue that's able to absorb the foliar spray. There we go. All right, so the last cation that I'm going to talk about here is magnesium. And so magnesium is at the center of chlorophyll. That is one of its most important roles. And without enough magnesium, you can slow down photosynthesis. And as you can imagine, that causes a cascade of negative consequences. Photosynthesis is how the plant is getting its energy. And then that energy is converted into carbohydrates, and it really drives all of growth. And so you're going to have some slowed growth. You can have issues with not having enough carbohydrates and not enough nutrients to put into fruit development. All kinds of things are going to not work as well. So the plant won't be able to defend itself in the same way. All of its functions will be somewhat inhibited. Luckily, magnesium deficiency is not common. And this goes back to what Karl was saying about knowing your geology. I don't have slides about it here, but that would be an interesting thing to look up, because there are some soils whose parent material, the rock that the soil is made out of, is low in magnesium. And then some that are high in magnesium, and either one can cause issue. Really what you want to do is take a soil test to see what your ratio of magnesium, potassium, and calcium are. All three cations compete for uptake into the plant for a certain extent, and you can add ammonium in there as well. Excess ammonium can cause potassium and magnesium deficiencies in some cases. So just try to keep all of these balanced. So in order to make sure you've got sufficient cation availability, like I said before, test the soil, test the irrigation water, and I should add in there leaf tissue, and look at the ratios between calcium, potassium, sodium, and magnesium. And pay special attention to crops with high potassium requirements like celery and avocado. I have seen studies showing celery to take up like 450 pounds of potassium an acre. And here in California and Ventura County, we're growing a whole lot of celery on the same fields year after year. So we want to make sure that we're replenishing that. Also, we want to maintain a healthy root system to facilitate the nutrient uptake. Like calcium especially is taken up just on those really like sensitive root tips. And so you need healthy growing roots in order to take up enough of these nutrients via the root system. And you want to continue supplying them throughout the season, because calcium, especially once it gets to its destination in the plant, it's going to stay there. So you want to keep on supplying it so that the roots have calcium constantly coming in. They could end up in the leaves, and then the roots may become deficient in one if they're not fertilized later on. And that could impact the amount of nutrition that that plant is able to take up once fruit starts sizing up. These nutrients are also required in particular times during development. So most of the calcium, for example, is taken up into the fruit in a relatively short amount of time. So looking at the resources like YAR's fertilization guidelines and UCANR's nutrient uptake curves will show you when you need the highest amount and the rate at which the plant is taking up calcium, potassium, and magnesium at certain times during the season. So consult your CCA. All right, so now we're gonna go on to the micronutrients. And these guys are involved in really everything. I'm just listing a few here. There's plenty more. Iron is required to produce chlorophyll. It's needed to activate enzymes. It's involved in respiration and photosynthesis, and it's a component of chloroplast membranes. Zinc is really notable for its requirement to produce auxins. That's a group of growth hormones. And if you don't have enough zinc, you end up with shorter spaces between nodes. You can end up with smaller leaves, rosetting, which is a clustering of leaves at the top of the plant. Chlorosis is very common, so I'll show more of that. It also is required to make tryptophan, which is important for protein production. Then, like the others, zinc is involved in enzyme activity and chlorophyll. Copper and manganese, that's involved in electron transfer, and so you need it to continue photosynthesis and respiration. Both of them are also involved in forming lignin, and the lignin is really important for making the crop resilient against an invading insect or pathogen, as lignin is difficult to chew through. It's also required to synthesize phenolic acid and other chemicals that help deter invading organisms. And both are also important to lipid structure. Boron, people are pretty familiar with that for its importance in cell walls and forming pollen tubes and seeds. And so we've got to make sure we have enough of that and calcium at the same time. And it's also important for moving water, carbohydrates, and nutrients around the plant, translocation. Okay. Yeah, so this is really just a summary of everything on the previous slide. And the photo here is of citrus, and that's a zinc deficiency. And a lot of the micronutrient deficiencies can look quite similar to each other if you're not familiar with them. Or like citrus, it's very easy to confuse it with HLB, and the two often go hand in hand. And in Ventura County, citrus trees very commonly get zinc and manganese deficiencies. So you'll generally know in your area which micronutrients to watch out for the most. You've got to make sure to get all of the micronutrients that are needed so that the plant can produce all the growth hormones that it needs, and so that it can initiate all of its defense mechanisms, from structure and lignin production to polyphenols, phenolic acids, and things that deter organisms. So I wanted to talk about nutrient availability, and I know that, oops, let me go back one. There we go. This is just a picture from a common textbook, and it's showing just how quickly micronutrient availability decreases with increasing soil pH. And you can see that nutrient availability is dependent on a lot of things: soil moisture, organic matter, chelating agents, and nutrient interactions, like Karl talked a lot about with the graphic in his presentation. The soil type affects it, and water quality. But if we take a look at this graph, we have cation concentration on the y-axis, soil pH on the x, and you can see iron exists in soils in two forms. And in a well-drained, aerated soil, iron 3+ is the dominant form. And you can see that by the time soil pH gets to a little bit below 8, there's basically no iron available. It's gone very low. And so you can see in this graph next to it, in order to meet plant demand, you've got to be at this line here. And then total solution iron is on this line. So the iron at a higher pH is is way below what would be required. So in order for a plant to get the iron that it needs, it's not going to be freely available in soil solution, because at the common soil pH, it's well below what the plant would require. So the only way we're going to get that iron is through complexation. So let's go to the next slide. So this is an example of a chelating agent, EDTA, in lots of fertilizers. And it forms a stable complex with a metal. So for example, iron or any of those positively charged micronutrients. That would be at the center, and it's held together by this organic molecular structure. And this guy is stable in a much wider pH range. And if you're familiar with EDDHA, that is stable up to, I think, pH 12 or something. So by including these in your fertilizer solution, you can make sure that the micronutrient is delivered to the plant root before it can precipitate out of solution. EDTA is not the only one. You can also get complexing and chelating agents from organic matter. So humic acids, fulvic acids, amino acids, like lots of different kinds of organic acid molecules form bonds of different levels of stability with micronutrients. And they're all very important for keeping all of the micronutrients in a plant available form in the soil solution. You know, you've got to make sure you also have good soil moisture, but having this by either including chelating complexing agents in your fertilizer or building up the soil organic matter can all help towards making sure that you can meet the crop's micronutrient demand. And then also doing foliar sprays is important as well. And with that, I would say go with a complexing agent that is maybe a little gentler than an EDTA, like a lignosulfonate or an amino acid complex for foliar spray. So just like so much in agriculture and soil science, soil health and crop health always comes back around to organic matter. Organic matter increases the soil's cation exchange capacity and anion exchange, meaning it can hold more nutrients, both positively and negatively charged. It increases water holding capacity so that the crop has access to more of that water for longer, and you're hopefully leaching less. Organic matter is also very necessary to create soil structure, and that structure will help the roots develop. Healthy network keeps it aerated. Good drainage keeps sodium moving down. It's important for proper transport of nutrients throughout the profile. It's important for healthy microbiome. Organic matter also buffers the soil pH. So if the pH is very high or very low, it'll bring it back closer to neutral. Organic matter also provides those naturally occurring chelating and complexing agents, like I said before, and it is created by and necessary for healthy microorganisms. And so that's a whole other presentation, but you've got to feed the good microbes, and then in return, their activity, their growth cycles are building up the organic matter store, stabilizing organic matter, and creating structure. And all throughout that time, their metabolites and activity helps make nutrients more available to plants. So I think that wraps up my presentation. Here's my contact info, and I'll open it up to questions.
Thank you, Erin. And just really quickly before we go into the Q&A, some people have been asking, hey, wait, are we recording this? You guys are going too fast. And so some people want to go back and watch this again, like a GoPro video or something like that on their mountain bike ride. So yeah, we are recording this. If you look up in the upper left-hand corner of your screen, you'll see the little red dot where it says recording. At least I see it on my screen. We are recording this, and I believe we'll talk to Miranda and Madeline and make sure that that is available for you on our website. So WRCCA.org, is that right, Madeline? Or Miranda?
What we'll do is at the end, we'll put our contact information in the chat. And if anyone wants the link, they can email Madeline and she can send them the link. It takes Zoom a little bit to render the video and make it available. So it probably won't be available until tomorrow.
Sounds good. Thank you. All right. We do have one quick question. Let's see. Asking about foliar calciums, do you have any preferences in terms of forms of foliar calcium, Erin, that you find to be more effective?
I hesitate to say. I think that it depends on the other micronutrients that you might want to include in that foliar spray. In places where there's a boron deficiency, or there could be a boron deficiency in the near future, it's good to include boron with your calcium. However, in my area, there are a lot of places with boron toxicity. So in that case, you want to really, really avoid it.
Mm-hmm. Exactly.
I'm not going to say any brands, but I would look for amino acid complexed micronutrients for foliar. Also, humic and fulvic acids applied foliar help with the uptake. And then there are also things like lignosulfonates that are some sort of organic complexing agent.
Mm-hmm.
EDTA, EDDHA, those are not the right fit foliarly because they form too strong of a bond with the micronutrient, and they can be absorbed into the tissue where they could release the original micronutrient, but then take up other micronutrients in the cell, wreaking havoc. So don't do that one.
Agreed, for sure. Yeah, it's difficult to make a recommendation as a CCA. Definitely, we don't want to play favorites too much. So no, I think that was a very good answer.
Thank
Okay,
you.
in the interest of time, I'm going to shut down the Q&A on this one. Thank you, Erin, very much. That was really good.
Thank you.
I've got a bunch to present as well, so I want to get my presentation going here. And let's bring it back up to the beginning. For my presentation, I would like to talk about something I've been talking about quite a bit, and that would be interpreting soil water analysis. So basically, we've got a lot more of these irrigation and nitrogen management plans, and that requires that you have a nitrate analysis of your irrigation water to put on your plan. And so that's been driving, we didn't used to get a lot of water analysis through the laboratories, but that's driven demand for water analysis right off the charts. But, it's really a boon to the industry to have people look at their water more intensively, because water is the single largest crop input that we have, and it contains a lot of ions, or salts, if you will, and I'll talk about that in just a bit. And once again, water chemistry has a big effect on your fertility program, and depending on how your water is, it can really have a big impact on what crops you choose to grow and what that final yield could end up being, and that would be your salinity management. So If you get a water analysis back, I mean, you're looking for just nitrate, but there's a wealth of information on that suitability analysis that hopefully this presentation will help you understand a little better. So I'm going to talk about pH in the carbonate buffering system, and I'm going to spend some time on it because it is important. We'll talk a little bit about EC and salinity and how that affects crop types and their sensitivity to salinity. Talk about cations and anions and what is typically found in water, how sodium can have an impact on infiltration. And then we'll look at quickly specific ion toxicities and then microirrigation concerns. There's certain things that can plug up your drip system. So let's get into it. So
Jerome,
go ahead. Yes.
Can we share your PowerPoint?
Oh, I'm just bombing along. I thought I shared my screen. What? Hi, I can see it. How come you can't see it?
Okay. So if you go into the Zoom interface.
There we go. Is that it? Share. Oof. Is that working now?
Yes.
No.
Yep.
Is that it? I'm sorry.
No problem. No
I was
problem.
just ranting away. Okay. Thank you. Sorry about that. Anyway, so here's a typical water report, a suitability water analysis. You know, that I'm familiar with working with Precision AgriLab when I used to be with Nutrien. Just a little while ago, I moved to another job, but this would be a typical report. I think this is a really good report. It gives you pretty much all the information laid out in a nice format. And the top part typically talks about pH, the salt or the salinity of the water sample, gives you the sodium adsorption ratio. So that's the balance of the sodium to the calcium and magnesium. Talks about hardness, alkalinity, and then whether or not you're going to get a scale formation, say like in a drip system or like on your shower head or something like that. The next section down below that talks about the cations, and then we look about the major anions and then look at the balance of those two things to make sure that the lab did a good job. And then finally, sort of those specific ions that I was talking about that could have impacts on basically toxicity to the crop, the boron, or the iron and manganese are pretty special things for your microirrigation system. So let's get into it. So the most important thing that people think about is pH, and it's kind of everybody knows about it, but I don't know how many people actually sit and think about it. But we all know that water dissociates very little bit, dissociates from just plain old water into protonated water and so an H plus or an OH. And that does have a big impact on our water chemistry. And so we do like to have a look at pH, and I'll tell you why that's important in a second. But you know, pH is the negative logarithm of the hydrogen ion concentration. It's a big mouthful. So typically the pH is just a shorthand of dealing with that exponent on the hydrogen ion concentration in this equilibrium reaction that happens all the time in water. So if we've got more protons than hydroxyl ions, we say the pH is, say in this situation, if it was 1 to 10 to the negative 5, we just say the pH is 5. We don't worry about the negative. We just look at the exponent. And that tells us a lot about that water chemistry. And anything below 7 is typically considered acidic. Anything above is considered to be basic. And neutral is right there at 7, where we've got a balance between protons and hydroxyl ions. But why is that really important? It mostly has to do with the solubility of the ions in the water. And really, it has to do with our salts and our fertilizer materials. You know, basically, how soluble are they? Certain things are affected by pH in terms of solubility, and other elements are not. My favorite example would be limestone or calcite versus gypsum. Gypsum is not really affected. Its solubility is not affected by pH. So depending, you know, the pH would have to get pretty high to have an impact on gypsum solubility. But we see with limestone that if you get above about pH 6.5, you're not really getting much solubility at all. We see a rapid decrease in the solubility of limestone as that pH goes above neutral. So, and there's a lot of other elements like phosphate can be very soluble if it's at an acid pH. Also, I think Aaron was talking about the solubility of the micronutrients. We all see those metals are much more soluble and much more available at a low soil pH. So pH has a big impact on the availability or the lack of availability of various nutrients. Also, if we're injecting fertilizer into water, we really want to know what's going to happen with that fertility, and pH has a big impact on that. You know, if we have a high pH irrigation water, we can get a big problem with ammonia volatilization, or losing that ammonia out into the atmosphere. We want to get it into the plants and keep it in the water, but you know, if the high pH water is going to cause a problem, you're going to smell that ammonia and you're not going to be getting it out to the plants. Also, if we have high pH water, we can precipitate calcium phosphate. So certain fertilizers, say like an ammonium polyphosphate or 1034O, if we have a very high pH water, we have a lot of calcium, you know, at that high pH, we're definitely going to get some precipitation. You can plug up a drip system pretty fast. High pH also will cause scale formation. I'll talk about that in a little bit more detail in a further slide. Most pesticides, it depends on the pesticide, but if we are water running pesticides, or even in our spray tank, it can have a big impact on the hydrolysis of that pesticide. If our pH is too high, we typically will buffer a spray tank with an acid to make sure that that pesticide doesn't get hydrolyzed. And we're talking about drip systems and things like that. We'll take a very high pH water in many cases, and I'll talk about the specific situations a little later. But if you have a high pH water that's, you know, over 7.5, 7.8, 7 or 8.0, something like that, we'll typically lower that water pH to an acidic form so that we can get the bicarbonate levels down. And sometimes we can lower the water pH to acidify a high lime soil and release some calcium and get our nutrients more available. So pH is quite important. It's not the only thing that you need to know about your water, but it is quite an important one. Now, what has a big impact on water pH is this carbonate buffer system. And a buffer is something that resists pH change when you add either an acid or a base. So basically, if you add a bunch of acid and your water is well buffered, there won't be much pH change until you add a whole lot of acid. So bicarbonate, carbonates, and carbonic acid are three elements that are all in equilibrium in any water system. And those things are what allow that water pH not to crash if you add something to it that could either acidify or raise that pH. So the more balanced or more buffered your water is, the more stable it is. And so waters that come straight out of the mountains, out of the Sierras, that are low in total bicarbonates or carbonic acid carbonates, they tend to have a rapid pH change with the addition of even a small amount of acid. But bicarbonate is usually the thing that we see the most in typical irrigation water samples. And we typically see bicarbonates dominate once we get up to about a pH of about 8.3. And then after that, carbonates are dominant. And quite often we see high levels of sodium at those very high pH waters. Here's the carbonate buffer system. And basically it shows that at low pHs, we have carbonic acid, which makes sense. So basically the bubbles in your typical soda would be your carbonic acids. Carbonates, though, in most of our waters, we rarely see a water that goes below pH 7 here in the West. Most of our waters range from neutral to, you know, up to high pH in extreme situations, but typically around 9. So we see that, you know, bicarbonate is our dominant species right there in the middle of the chart. So that's our big player, and that keeps that, if the bicarbonate levels are fairly high, that will keep that pH from changing too much. But sometimes that's not good to have too much bicarbonate in there because it can cause issues. I'll talk about that in a little bit. But if we're talking about well water, almost all well water has some sort of bicarbonates in them. But where does it come from? Well, typically we have a lot of limestone in our soil, naturally present in these young soils out here in the West. And rainwater typically absorbs carbon dioxide. There is carbon dioxide in the air, and apparently more and more every year. But the carbon dioxide that's in the air is absorbed by the rain. And so rainwater is naturally slightly pH, has an acidic pH of about 5.7. And as it percolates through the soil, in this equation at the bottom of my slide, it takes that limestone on the left with carbonic acid and releases calcium, and you end up with bicarbonate. So typical waters in an aquifer are usually at an equilibrium such that we have a bunch of bicarbonate in the water. So usually the deeper the aquifer and the older the parent material, the more bicarbonates we have in there. So once again, should we be concerned about excess bicarbonates? Well, because they can tie up certain micronutrient metals. If we have bicarbonate levels over 2, that would be about 2 milliequivalents per liter, about 122 part per million bicarbonate in your sample. That may cause scale formation. So basically the reverse of the reaction that we just saw on the previous slide. We basically form calcium carbonate, precipitates out when the bicarbonate reacts with the calcium in that irrigation water, and we end up with that calcium forming a hard mineral that is very hard to dissolve. And interestingly enough, this is one of those things, most salts go into solution faster at high pH, at high temperatures, excuse me. But in the situation with this reaction, as the temperature goes up, we tend to form more lime and more scale. And that's a real problem with that black hose. It typically heats up quite well when it's exposed to sun of our Western weather out here. When we have bicarbonate levels that are about 3 milliequivalents per liter or more, we start to see precipitation of those metal micronutrients that are so important for crop production. You don't need many, but what you need is quite important. So iron, zinc, manganese, and copper, as Aaron was showing, will tend to precipitate out at those higher pHs, and they react with bicarbonate and settle out, and they're unavailable for plant uptake. Also, bicarbonates precipitate calcium, magnesium, which are beneficial cations. And that means in our SAR equation, which is a ratio of sodium to calcium and magnesium, basically you're removing calcium, magnesium from the irrigation water, and that makes the sodium even more dominant and causes even more problems. And once again, as we're adding this bicarbonate to our soil, we get this liming effect, and that tends to raise your soil pH to the point where we can have mineral tie-up or nutrient tie-up. So there are variations in aquifer chemistry. Typically on the east side of the San Joaquin Valley here in California, we have very pure snowmelt type water. We have granite soils that have very little limestone in them. They're made from quartz. And so we have very little bicarbonate in our eastern waters. Even some waters east of Highway 99 are almost like a snowmelt water. They have very low bicarbonates in them, very low salt contents, even in well water. And so soils that are more on the coastal side of and right along the coast, high in limestone, you can have a great deal of bicarbonates to the point where they can be quite detrimental to crop production. So there's this calcium saturation index. That's a value that you see on a lot of water reports. And that's an equation that's run to determine whether or not scale is likely to form from application of irrigation water. Will you get those deposits forming on the side of your glass or in your drip system? Can you plug up drip emitters just through scale forming alone? And this Langelier index is just a way of taking your pH value and then with a calculated pHC value, which is based on concentration of calcium and the bicarbonates in your water, also the minerals in your water, your calcium, magnesium, sodium. You crank that number and you look at the balance of those two. So if that Langelier saturation index value is positive, then it's quite likely you're going to get some scale formation if you use this water through a drip system. If it's negative, then what's interesting about that is that water, typical of snowmelt water, will have a negative saturation index that it will actually start to dissolve calcium out of your soil and leach it, remove calcium from the system. We call this water corrosive. And so basically at that point, you definitely need to add more calcium to your water so that you don't run into a calcium deficiency. It'll leach that calcium below the root zone and you end up with very calcium deficient topsoil. So let's talk about alkalinity and hardness. Specifically, those are two different things and people can get them confused. Alkalinity specifically refers to the anions, the bicarbonates and the carbonates that are in that water. And once again, those things are in an equilibrium depending on the water pH. So if you have a water pH that's 8.3 or less, you're going to have mostly bicarbonates. But if you're, say, down in the southern end of the valley or in the desert, you can see water pH is above 8.3 and you actually start to get carbonates. And those can be get to up to a toxic level in some cases. But that's what we're talking about in terms of alkalinity. In hardness, that is specifically looking at the minerals in your water, specifically calcium and magnesium. And if you have very high values of calcium, magnesium, or hardness, that can lead to scale. And it also can precipitate your phosphate fertilizers if you try to inject those into a high hardness water. So anything over about 140 part per million hardness, you probably should avoid injecting 1034O into a drip system. And you're going to want to go with more of an acidic form of phosphate. So what do we do about high bicarbonate water? If we've got a bicarbonate level that's above 2 to 3 milliequivalents, 3 milliequivalents is where we start to get really nervous. And so if it's in that low zone, it's kind of right on the edge between 2 and 3, you can typically go in maybe on a drip system or like a sprinkler system that it's based on PVC, like some of those micro sprinklers, something like that. You can go in with a little bit of anfuric and treat that about 3 to 5 gallons per acre, lower that pH of the water down to about 4.5. Do that maybe once a month or every couple of weeks. And that should keep everything clean, remove the bicarbonates from around the drippers, and also sweeten your soil around the dripper as well, where the bicarbonates tend to build up. But a lot of our irrigation waters on the west side can have very high bicarbonates, above 3 milliequivalents per liter. That's going to be about 180 part per million of bicarbonate. And those frequently require continuous injection with a sulfuric acid system. And the common economic level to lower your water pH in those situations would be down to between 6 and 6.5 downstream of the injection point. And if we do that, we're going to lower our carbonates by about 50%. So you can take a bicarbonate load that's 3 milliequivalents, lower your water pH to about 6 to 6.5, and about half the bicarbonates will be gone. It'll just gas off as CO2, and you have a bicarbonate level of about 1.5, which is really pretty nice. There are certain situations if you have high lime soil, let's say like some labs reported as high fizz, or some will actually calculate the lime percentage of your soil. You can actually increase the sulfuric acid injection rate and not only treat your water, but actually treat your soil a little bit. There'll be some residual sulfuric acid left over, and that will actually begin to dissolve some of that limestone and make that calcium available and make your nutrients available as well. But the thing is, people will ask, okay, the water pH that I have is X. How much acid do I need to inject? And due to the complexity of water chemistry, there's no way to really guess that. There are some tables that you can try, but the best thing to do is to ask your testing lab to run a titration analysis with sulfuric acid or another acid, phosphoric acid or anfuric acid, or other alternatives. They can do those titrations for you and tell you exactly how many gallons you need to run to achieve a final pH. So moving on from bicarbonates, pH, and all that, we'll talk about salinity and its effects on crop production real quickly. So water, pure water with no ions in it, so deionized water, does not conduct electricity very well. But as we get positive and negative charges in that water, the conductivity of that water increases as we get more and more ions in there. So we used to measure the total amount of salts in a water by just evaporating that water sample on a watch glass and then weighing the salt that's left over, but that's total dissolved solids. It turns out that that's a pretty inconvenient way to measure the number of salts, the amount of salts that are in your irrigation water sample. So we just look at the concentration or the conductivity of the water to figure out what the concentration of our salts is. And there is a relationship between total dissolved solids and the conductivity of your irrigation water. And you just multiply by roughly 640 to calculate total dissolved solids if all you have is your conductivity value. But typically conductivity or your EC is what we need to know, because that's how a lot of our sensitivity to crops to salinity are calculated. We've got lots of tables based on EC and not so many on TDS. So TDS is kind of an old value. That's an old school lab that reports that. Sometimes they'll give you both. What's an interesting fact is if you know the conductivity of your irrigation water, which is commonly reported, all you have to do is multiply that by 1.5 to estimate what the salinity of your soil is going to be if that soil has good drainage and is at a steady state. So I always say that your soil takes on the chemistry of your water. Well, basically, due to evaporation and the concentration of salts in the soil with good drainage, typically you're going to get about a 50% increase in conductivity of your soil if you use that water over time. Now, if your conductivity of your soil is higher than that relationship, that 1.5 times the water conductivity, that means you probably do have a drainage problem, and you need to investigate further to see if there's something impeding the downward flow of that water. You probably don't have good drainage, and you're starting to get a buildup of salts in the root zone, and that's something you do need to take care of. So why is salinity so important? Why do we measure that electrical conductivity? And it all has to do with the fact that plant roots need to expend a lot more energy to take up water from a soil solution that has higher and higher ions in there. So plants are all different. There are certain plants that do really well with higher levels of salinity. When we see high salinity soils or irrigation water, we typically want to recommend a crop growth like a pistachio, or there's sugar beets or barley can handle very high levels of salinity, versus say almonds or citrus, onions, lettuce, they like low levels of salinity. And so I just want to mention here that sometimes people get salinity and salts confused. Sometimes they think of salt as table salt, and they think that salt, when I mention salt, I'm talking about sodium problems in particular. But when I'm talking about a salinity issue, I'm talking about all the ions in that water, everything in that water that's not water. So basically all the ions, calcium, magnesium, all the anions, added all together, that's what we're talking about in terms of salinity. And it's just an osmotic potential thing that really limits crop growth. Certain plants do better and certain plants don't. There are tables out there that are available. There's extensive information on the internet. From the FAO has a really good publication that lists all these the crop sensitivities to salinity. And there's a specific relationship, a linear relationship that says, our relative yield. So if we're doing a good job on everything else and salinity is our only factor, that crops can usually reach a certain threshold of salinity before we start to see a yield decrease. So the threshold level is that A value, and then the slope is what happens as our salinity increases further. And let's show an example of sensitive crops, say like an onion, lettuce, and maybe almonds on certain rootstocks, are more sensitive. And you can see that you don't have to get a very high salinity level in soil before we start to see crop yield reductions. And then we get out to very tolerant ones, can take an electrical conductivity of a soil solution extract. You can get out to 10 before you start to see any reduction in yield. So that would be something more like date palms or pistachio can handle a great deal of salt. So I'm talking about soil values, but it's all tied back to the water salinity. And it's very important to know what your water salinity is because your soils will take on the characteristics of that water. If we do have excess salt, knowing what the leaching, what the limits are for that crop are very important for us to be able to calculate how much extra water we need to put on to be able to remove those salts from the root zone. So assuming you've got really good drainage, basically you can use this equation where we have the electrical conductivity of our irrigation water in the numerator, and then out of those tabular values in the denominator, you pull that out of the table for what the crop requirement is, and it gives you a fraction of extra water that needs to be applied to make sure that we're leaching salts below the root zone and that crop can continue to grow even though we have salinity of water that may be a little bit higher than what that crop really would like to see. So leaching is the answer, but we have to make sure that that soil drains well if we're going to take advantage of this effect. I'm going to jump to the cations and anions, and those are in tabular values, usually on any irrigation water report. Sometimes they're reported in parts per million, sometimes they're reported in mill equivalents per liter. There's definite advantage of reporting them in terms of mill equivalents per liter because we're taking the charge out of the equation and just looking at how the balance of positive charges to negative charges is. And if the lab is doing a really good job, if you sum all your cations in terms of mill equivalents per liter, sum them all up, sum up the anions, they should be equal. And if the lab has missed something or there's been a miscalculation and those things don't equal each other, then we know that you should probably ask the lab to rerun the sample or find a different lab. It's very rare that we see this, and most labs will show the balance of the cations to anions on their reports to show the quality of the data. And one other thing that's kind of interesting is if either the sum of the cations in terms of mill equivalents or the sum of the anions, we know they're equal. If you multiply the EC by 10, you should be able to determine what that sum of the cations or anions is. Or likewise, you take the cations summation, divide by 10, and it should be equal to the electrical conductivity of the water sample. Another quick check to see, is the lab doing a good job? Did they miss something, or do you need to send the sample back? I'm going to jump to sodium problems, and mostly this is something to do with infiltration problems. Sodium has a strong positive charge, and so it tends to attract a big shell of water, which ironically dilutes the charge density of that very dense positive charge. So sodium is very loosely held to clays and tends to make clay particles disperse. So they can't really bind up to each other. Negatively charged clay particles will repel each other. And what happens is they tend to plug micropores, usually at the surface of your soil solution, of your soil surface. Calcium, magnesium have very large positive charges. They have 2 positive charges. There's a lot of room for clays to bind to calcium and magnesium, and they can flocculate clays and provide channels for water to move into the soil. And typically this is happening in the surface layer, say maybe 1 to 2 inches of soil. We take a sample usually to 12 or 18 inches, and we're not really capturing the calcium that can be lost in that top layer. So that top layer can be dominated by sodium, and you can get calcium leached out, and you can lead to real water infiltration problems if sodium is an issue. So this is that SAR value that is reported on most soil tests. And once again, it's basically a ratio of the bad guys, sodium, to the good guys, calcium and magnesium, in the denominator. And there's some values out there. Basically, if your sodium absorption ratio, basically sodium is dominant. If we get that value over 3, we're just beginning to see problems with water infiltration. So you put a sprinkle application out and the water tends to pond up and it just won't go in the ground. If we get above 6, we're starting to see real problems, and above 6, the water is just going to sit there. It can sit there for days. Commonly you can see irrigation, if you go out into an orchard or something like that, where you've got a dripper and you see algae or moss growing on the soil, it's probably a sign that you have water penetration problems. So Adjusted SAR I talked about is a situation where we have high levels of bicarbonates or carbonates in our water. And those bicarbonates, once again, are a problem because they're absorbing or binding up the calcium, magnesium, which are beneficial elements, and allowing that sodium, which doesn't really precipitate with bicarbonate. It forms basically baking soda, which is soluble at just about any situation. So that sodium tends to dominate if we have a lot of bicarbonate in there. It makes calcium and magnesium go away. So that SAR is a very important term that you see on your irrigation water report. But if we have some sodium present and we also have very low salts in general, if we have very pure water coming out of the Sierras and it doesn't have calcium and magnesium in it, but it does have sodium in it, then we have kind of the worst situation possible. So low EC water plus high sodium leads to the water almost won't go in the ground at all. And I've seen even very sandy soils that have just a tiny bit of silt in them, just enough to plug things up, even though the water is essentially what everyone calls good water. It's almost too pure. And so we do need to add calcium into those waters, say like a gypsum injection or calcium liquid supplements, just adding some sort of salt to raise the conductivity of the water up so that we can begin to flocculate some of those silts and clays and get that water to infiltrate into the ground. So treating high sodium water, as I mentioned, we want to counteract that sodium, the bad guy, with calcium, the good guy. And the typical thing to do would be to use gypsum injection. And that's, we can put quite a bit of gypsum in the water. You can use liquid sources as well, although they can be quite expensive. But if you need some nitrogen, you can use P17, and you're going to get some calcium along with your nitrogen. Also, soil surfactants can help with breaking that surface tension. I'm going to skip along because I'm starting to run out of time. I'm definitely past my time here. But so if you're looking at your water sample, you want to see some ideal cation balances to make sure your water is in balance. You want your calcium to be about 2 to 5 times your magnesium. If we get too much magnesium in our irrigation water, it's possible to find things, soils that are derived from serpentine parent material and have high magnesium, low calcium. We want to make sure that our calcium is dominant, and some of these values are recommended. I'm going to skip along. Our common anions in the water would be our bicarbonates, chlorides, sulfates, and nitrates. Once again, our drinking water standard in terms of nitrate as nitrogen is 10 part per million. And so we definitely want to make sure that we are taking it into account our nitrate nitrogen. That's what we're reporting on those INMP reports. Sulfate in irrigation water, really, we don't have to usually worry about that too much unless it gets very high. Over 400 part per million can cause some real issues, but it's rare to ever see that. Typically, most crops can handle a good amount of chloride. A lot of people freak out about chloride, but most crops are sensitive. Even the sensitive crops can handle about 250 part per million, or say up to 4 to 5 milliequivalents of chloride in irrigation water before we start to see real issues. If you have a drainage problem, then the chloride can build up and we have real problems. And I talked in depth about bicarbonates. We definitely want to stay below 2 milliequivalents if we can. Talked about chloride toxicity. It is the most common one out there. The perennials are the ones that really suffer from chlorides the most because, we don't plow them under. They tend to build up in the woody tissues. And so, 250 part per million, or about 7 milliequivalents per liter in the water, I would say is about the upper threshold. And we definitely want to keep most leaf tissues under about 0.3% chloride by weight. Different rootstocks can handle different amounts of chloride. But the thing is that you can't really get chloride out of your irrigation water. You can't remove it practically. So the only way to leach, to get rid of chloride would be to leach it. And so chloride leaches really well. It's very mobile in soil. And so a little bit of excess water will help remove chloride from your soil and keep it below the root zone so we don't have it build up. But if you have a drainage problem, you're definitely going to want to take care of that because then you can't get rid of that chloride. tiles may be necessary to improve drainage to get that chloride out of the root zone. Boron toxicity, we do see that in certain areas, usually from some of the geothermal or seawater mineral type sources, Cache Creek, Woodland, Tracy, those are areas that are known to have high levels of boron. In irrigation water, up to about 1 part per million boron is typically beneficial. Somewhere between about 0.5 to 1 part per million is pretty good. But when we get above 2 part per million, we start to see toxicity. The thing to know about boron is that it's much harder to leach out of the soil. So if you do have high levels in the soil or in your irrigation water, it will tend to build up in your soil pretty quickly and you can have some real issues. So it takes about 3 times as much water to leach boron as it does chloride. And finally, iron and manganese levels, even a small amount of iron or manganese in the presence of iron bacteria or manganese reducing bacteria can start to cause a plugging in micro irrigation systems due to the formation of these slimy filaments. And you see those if you cut open a dripper and you feel that slippery kind of orange or black material in your irrigation dripper. That is usually a sign that you've got some iron manganese in there that is growing those bacteria. And we have to kill the bacteria, and we can oxygenate the water to try and precipitate the iron magnesium out of it. It can be very tricky, but even 0.1 part per million of iron or manganese in your irrigation water can be enough to cause problems. So Netafim USA has a really good paper on how to control iron in a drip system, and there are different strategies you can do, but usually chlorination, if the iron levels are quite high, continuous chlorination may be necessary. But once again, if you have the ability to oxygenate your pond and precipitate the iron and manganese out before you pump the water into your irrigation system, that usually can be pretty effective. Finally, there are certain labs that will do a dip plate that will check to see if that bacteria is present. And if you do have bacteria, it can grow in those drippers, and algae can grow as well. Chlorination is definitely the way to treat those, but you can look at the presence or absence of number of colonies in your irrigation water sample to find out if you're going to have a problem with those types of elements. Typically, surface waters are going to have more of these, but you can have shallow well water that can have these iron-reducing bacteria and can cause real problems with the drip system. I've seen it in certain situations. So finally, this is when you get your irrigation water sample back. Most labs will do the calculations for you, but if you do need to make some, do some things and calculate the number of pounds of any particular element per acre foot of water applied, basically the magic number is 2.72. That number can be used to multiply by anything in part per million to determine how many pounds per acre foot you're applying. And then if you need to convert from parts per million to equivalent weight, so our mill equivalents, you basically just multiply by the valence of those. And I reported those previously in my presentation. Nitrate nitrogen versus nitrate, there's a calculation there. And basically, I'll just leave this on the slide. And when you guys need this information, basically just download this from our website, and all the rest of those calculations will be right here. So we've got some examples on how to go through and do those calculations. So you've got a big influence of the irrigation water on just about everything we do in terms of fertilizer injection, pesticide chemistry, and all that. So make sure you get your water analyzed, have a look at all those chemistry things that are available for you. Don't just look at the nitrate, pick that value off, look at the rest of it and understand how that water can affect your production. So that I'm definitely way over time on my presentation, and my bad for that. I would definitely look at some questions. Let's see. What do we got here?
So we have a couple questions. The first
Good.
one is, have you experienced or have you thoughts on the use of the continuous applications of sulfuric acid chemistry in irrigation water being detrimental on soil?
Not really if it's adjusted right. If we need that sulfuric acid, I've never really seen a problem. Sulfur, people can maybe get a little bit nervous about having high levels of sulfate in a soil sample, but sulfate is one of those crazy things that it's very hard to see a relationship between sulfate in the ground or a soil sample and sulfate in plant nutrition. I've seen situations where we've had excess sulfate in a soil sample and still have a sulfur deficiency. And sometimes we see high levels of sulfate, even though there's usually not much sulfur in the ground. It can happen both ways. So there's very poor correlation between that. That's one of the few elements I would say would be sulfate, sulfur, and the sulfate that we find in plant tissues. So I've never really seen a problem with it. If you need the sulfuric acid, I'd say put it on. And it really is more about the bicarbonates and making sure that we keep our calcium available and we don't tie it up. That's much more important than worrying about the sulfuric acid. Now if somebody hits the switch and puts too much sulfuric acid on, maybe you could see your pH drop. But I think you want to do annual testing, especially if you're on a drip type system or something like that, to make sure that we're not overdoing it. Okay, next question.
How does a grower simultaneously implement a leaching fraction for salinity mitigation while at the same time practice good nitrogen management that avoids leaching nitrates and satisfies the water board regulators? This seems
so
to
that's an anonymous attendee, probably somebody from the water board that's trying to trip me up, huh? So basically, no, that is a very, very tricky thing to do. I mean, the obvious thing to do is to pay really close attention to your nitrogen management plan over time and make sure you're not over applying your nitrogen, and especially towards the end of the season. Understand how that nitrogen is being taken up by the crop. What you want to do is you want to run out of nitrate in the soil as you're getting to harvest. And that's pretty common anyway. You don't want to be continuously fertilizing as that crop is starting to run out of demand. If we have high levels of nitrate left over at the end of the season before the rainfall happens, that nitrate's going to end up in the groundwater, no doubt. So if our drinking water concentrations are 10 part per million, that's our threshold in terms of nitrate nitrogen. So I would say if you can get down 10 part per million or below, that's a pretty good value, to be shooting for. So don't overdo it on the nitrogen, and I think you'll be okay when the rains come and you start leaching to try to clean up your soil for next season. That's the best strategy I can think of. And so just paying close attention to nitrogen management will get you there.
So the next one is one needs to watch sulfuric acid amendments in core and other soil.
Exactly, yes, absolutely. So typically one is going to be very careful with sulfuric acid in a greenhouse or potted media type situation. Potted media is a whole nother presentation. And I was only talking about mineral soils in my presentation, and definitely, people are looking at other forms of acid to acidify irrigation water for potting media. Totally different situation. That's a whole nother topic of conversation, but good point. Thank you. All right. The moment you've been waiting for. Now in the chat, right, we've got where do we have?
Okay.
Our QR code.
So Madeline's
Stop sharing.
Yes, Madeline's going to share the PowerPoint with the QR code. I am going to put a bunch of links into the chat for everyone. So get ready for that. So it has all of our speakers' contact information. If you have more questions about their presentations, please feel free to reach out to them directly. As Jerome mentioned before, we have recorded this session and we can provide you a link to the recording after it's ready. To do that, you will need to email Madeline Shankin. Her contact information is also in the chat. And then the QR code is now on screen. If you have the CCA app, this is now the time to use that and scan.
I did it already. It's all right. I already got credit for it.
Yes, and so that's the fastest way to get your credits. If you don't have the app, you have to to wait for ICCA to audit our attendee list, and then they manually upload the credits to your account. So using the QR code is by far the fastest. We will also be hosting 2 more webinars. Each will be 2 hours, same time frame, 1 to 3 p.m. Completely free to CCAs on November 30th and December 14th. The links to those to sign up are also in the chat. And then I'm assuming if you're here, you probably already are receiving our CCA updates, but if you know another CCA who is not, we've provided a link to get those CCA updates on a regular basis. So whenever we're hosting webinars or have information about exams or specialty exams, we send those out via a newsletter essentially, and the link to sign up for that is in the chat as well.
Thank you to
So-
all our speakers. Really appreciate everybody staying on time except for me. And yeah, great job, guys.
Okay, so basically that concludes our meeting. I'm gonna go ahead and leave this last screen up and the webinar open for about 10 more minutes so everyone can scan the code if they need to, or copy and paste any of the links if they want. But at this point, the meeting is done, and I thank you everyone for attending. We know your time is valuable and we appreciate that you are spending it with us.
Thanks a lot, everybody. See ya. Thank you,
Thank
Erin.
you.
Thank you, Carl.
Thank you, Jerome.
All right.
Bye.
Bye-bye.